Okay, can everybody hear me and see me? Can I get a thumbs up or indicate in chat if you have audio? Okay, good, thank you. So today we're going to continue with chapter 40. Before that, a couple announcements. So remember, quiz number 18 is going to be today. I should have that posted on eCampus by about noon or so. Also remember that test number 3 is scheduled for next week. That's Wednesday, let me see what day is Wednesday. Wednesday the 18th is going to be test number 3. Keep an eye on eCampus and your email. I should have a study guide posted for you sometime later this afternoon. Again, Monday the 16th is going to be a review session. As usual, please look over your study guide. Please either come to the Zoom session or the class with questions ready. Again, I don't mind giving you these review sessions as long as you take advantage of them, as long as people are actually showing up and asking questions. Okay, I think that's about it as far as announcements go. If there's no questions on that, let's get back to chapter 40. Okay, so when we left off, we were talking about different strategies different organisms have for excretion of nitrogen waste. Okay, in fish, it's very simple. They simply breathe out diluted ammonia from their gills. In land animals and amphibians, however, there's a problem. We can't secrete, or excrete rather, we can't excrete diluted ammonia because that would mean too much water loss. Okay, we can't excrete concentrated ammonia because that means that it's too toxic. It would cause burns and damage if we were to try to excrete concentrated ammonia. Okay, so we have to, land animals and amphibians have to convert that ammonia to a less toxic molecule in order to excrete it. In the case of mammals, we excrete it as urea. So urea is excreted into urine in the kidneys. It's less toxic. It can be stored in the body for a while until we excrete it later. Land snails, insects, birds, and reptiles, they excrete their ammonia waste, their nitrogen waste as uric acid, okay, which is different. It's a solid white precipitate. Okay, so that brings us to homeostasis. Okay, homeostasis. Homeostasis is a state of physical and chemical balance or equilibrium within an organism. Okay, so in other words, homeostasis is keeping constant conditions within an organism. Some examples: temperature balance. Okay, so our body temperature should be around 32 degrees Celsius or so. Okay, 32 degrees Celsius. We need to maintain that constant temperature. Okay, so we do that by our circulatory system is one aspect of maintaining our body temperature. Okay, so warm circulating blood helps to keep a constant body temperature. If we get too cold, we might shiver. Okay, that shivering generates heat, again, in order to maintain body temperature. If we get too hot, we might start perspiring. Okay, the evaporation of sweat on our skin cools our body temperature down, brings it down. So this is all homeostasis with respect to temperature, keeping a constant temperature. pH balance. So you may recall from lab number 5, when we blew carbon dioxide into the water that contained phenolphthalein and sodium hydroxide, the pH dropped, okay? The carbon dioxide in our blood forms carbonic acid and bicarbonate. Okay, bicarbonate is a buffer. Okay, buffers keep pH at a steady level. A good example of this, so our blood, if we look at the pH of our blood, it's right about 7.4. Okay, and because of this bicarbonate buffer, it doesn't vary very much from 7.4. Okay, if it gets a little high, the bicarbonate buffer is going to start absorbing protons and bring it back down. If it gets a little low, the bicarbonate buffer system is going to start donating more protons and bring it back up to where it's supposed to. Wait, I got that backwards. But you guys get the idea. So if it gets too high, donate protons to bring it down. If it gets too low, absorb protons to bring it back up. Okay, that's the way it goes. All right. If our blood pH gets to like just a little bit higher, like 7.6, 7.8, that's a condition called alkalosis that can make you very sick. You know, possibly it could be even fatal. If our blood pH gets too low, like 7.0, that's pretty bad. That will make you feel actually very, very sick. That's a condition called acidosis. So we need to maintain pH within a very narrow range. That's homeostasis with respect to pH. Also, water solute balance. Again, like I mentioned on Wednesday, if our cells are in a hypotonic condition, that means there's more, the inside of the cell is hypertonic to its environment. Water is going to move down its concentration gradient by osmosis into the cells. That'll cause the cells to swell up, and they could possibly even burst. Okay, obviously, that's a bad situation, right? The opposite's bad too. If we put cells in a hypertonic environment, water is going to move down the concentration gradient out of the cell into the hypertonic fluid. This is going to cause the cells to shrivel up. Again, that's a bad situation. So organisms need to maintain a balance between water and solute in their body fluids so that their cells neither swell up or shrivel up. Okay, we need to keep the body's fluids isotonic to the inside of the cells. Okay, just like other things we've talked about, like different types of skeletons or elimination of nitrogen waste, different organisms have different strategies for carrying out osmoregulation, or homeostasis, in other words, with respect to water and solute balance. Okay, so let's start with the very simplest strategy for carrying out osmoregulation. We see that in marine invertebrates. So first of all, let's talk about this term. This is probably a term you've seen a lot, but might not know exactly what it refers to. So when I refer to marine invertebrates or marine bony fishes, exactly what kind of organisms am I referring to? What type of fish are marine fish? What type of invertebrates are marine invertebrates? In the ocean, Stephanie says, exactly. Okay, so these are saltwater animals. So marine invertebrates or saltwater invertebrates are osmoconformers. Okay, so they have a very simple strategy for osmoregulation, and that's to keep their body fluids isotonic to the water in their environment. Okay, by keeping their body fluids isotonic to their environment, they neither lose nor gain water. Marine bony fishes, on the other hand, are osmoregulators. So osmoregulators, body fluids are hypotonic to the environment and constantly lose water. To compensate, they constantly drink. Excess salt is pumped out through the gills. Okay? So again, because these are marine animals, so they live in salt water, Okay, salt water obviously has a lot of solutes. These solutes make ocean water hypertonic. Okay, in other words, having more solute than, let me get something to write with. Having more solute than the body fluids of the fish. Okay, so let me draw your attention to the camera. So if we have, again, pardon my artwork, I don't make a claim to be a great artist. So here's a fish. This is the marine fish. So its environment is hypertonic. To its body fluids. Its body fluids are hypotonic. Because its body fluids are hypotonic, water is going to go down its concentration gradient out. Okay, so we have a higher concentration of water in the fish's body fluids than its environment. That means it's going to lose water by osmosis. Water is going to go down its concentration gradient out of the fish's body. Okay, to compensate for this, the fish constantly drinks. It constantly drinks water in order to compensate for that water loss. It pumps excess salt out of its body, and very rarely do they urinate. Okay, marine birds and reptiles are osmoregulators. They drink seawater, and they have this special nasal gland that eliminates salt through the nostrils. Okay? Now, if you've ever seen a movie where somebody is maybe stranded on a desert island or stranded at sea where they have no source of fresh water, you know, they always say, you know, don't drink the seawater, don't drink the seawater, okay? And always somebody breaks down, does something stupid and drinks the seawater, and you know, then that person goes crazy and dies. We're not supposed to drink seawater. The reason for that is seawater is hypertonic to the cells that line your digestive tract. So in the digestion chapter, I told you that we gain water by osmosis because the brush border cells usually have a higher concentration of solutes than what's in the lumen of the small intestine. So water is absorbed by osmosis. Okay, if we do something like drink seawater, that's going to make the lumen of our gut hypertonic to the cells lining our digestive tract, the brush border cells. That means water is actually going to go out of the cells that line our intestines and into our gut. So actually, by drinking seawater, you're losing more water rather than gaining water. All right. However, some animals like seabirds and like marine reptiles, like the Galapagos iguanas, they can drink seawater just fine, but they have these special glands that remove excess salt from the water, and then the excess salt is secreted out of their nostrils, you know, from their nasal glands. Okay, now sharks and bony, pardon me, not bony fishes, but sharks and other types of fish with a cartilaginous skeleton, like sharks, rays, what else are there? Skates. Okay. These are osmoconformers. They're kind of a special situation. They are actually slightly hypertonic to the environment because they, instead of excreting their nitrogen waste, they accumulate it. This makes their bodies slightly hypertonic to their environment. They constantly gain water by osmosis. Okay, being slightly hypertonic, they're always going to be gaining a little bit of water by osmosis. So because of this, they don't need to drink, and they do urinate frequently. Okay, so again, kind of a special situation. Okay, so finally we have freshwater animals. In freshwater animals, the body fluids are hypertonic to the environment. Okay, so we have, this is for marine fishes. For fresh fishes, we have the opposite situation. Freshwater doesn't contain solute, doesn't contain salt the way that seawater does. That means that the water is hypotonic to the body fluids of the fish, which contain more solutes. That makes them hypertonic. Because of this, freshwater fishes are constantly losing water to the environment. I'm sorry, they're constantly gaining water from the environment. So water is going to go down its concentration gradient from an area of high concentration rather. Yeah. I just confused myself. Okay, so we have a high concentration of water, low concentration here, so it's going to go down its concentration gradient into the tissues of the fish. Okay, so they're constantly going to be gaining water by osmosis. Because of that, they don't drink. And to get rid of the excess water that's constantly entering their body by osmosis, they urinate very frequently. Okay, so that's freshwater fish. All right, osmoregulation in land animals. Land animals tend to lose water by evaporation and other processes. So the challenge is to osmoregulate, but not to lose too much water at the same time. Okay, so let's look at, whoops. So you might be wondering why there's a picture of this little rodent here. This guy is a kangaroo rat. And these are kind of unique animals. Kangaroo rats are desert animals. Obviously in the desert, there's very little water. These animals actually get almost all of the water they need from their diet, because in the desert environment, there's very little water around to drink, and they actually don't need it. They can actually absorb enough moisture just from the food that they find in the desert. Okay. The way that they do that is the structure of their kidneys is kind of unique. We'll talk about how kidneys function in osmoregulation a little bit later rather. But the kidneys of the kangaroo rat have the longest loop of Langerhans in their nephrons of any other animal on Earth. Okay, and this is important because those are the structures that are involved in reabsorbing water from urine. Okay, and we'll talk in a lot more detail about how that happens a little later in the chapter. Okay, so that's how the kangaroo rat deals with osmoregulation. So it has a very specialized kidney to prevent too much water loss, which is important because it lives in a desert environment. Okay, so how do animals gain or lose water and solutes? Water gain, absorption from the gut and metabolism. So we just talked, did a whole chapter on digestion and absorption. So we know that water is absorbed through the walls of the small intestine, through the walls of the large intestine. Metabolism. So when we think about aerobic respiration, remember that when we use oxygen as a final electron acceptor, we make a molecule of water. Okay, so we're actually, by metabolizing by aerobic respiration, we're actually making water. That's known as metabolic water. So we actually, it's not enough to sustain us, but we actually do make our own water, a small fraction of the water that we need. Water loss, urinary excretion is an example of water loss. Evaporation from the lungs and the skin. Sweating, okay, in some species, so mammals, well, humans, I should say, there are some mammals that don't sweat, but humans, we regulate our temperature by sweating if it gets too hot. Of course, other animals don't do that. Animals like dogs, you might notice when it's hot, dogs pant a lot because they don't sweat. They cool themselves by panting. But in some species, sweating is used to modulate temperature, and of course that results in water loss. Also, although our large intestine reabsorbs water from the waste material, we can't get it all out, so we do lose some moisture in feces as well. Pardon me. Okay, so how do we gain or lose solutes? Solute gain, we gain solute by absorption from the gut. Cell secretions, such as mucus, saliva, etc. Cell waste, nitrogen waste, we gain solutes that way. Respiration, O2, CO2. So when, by breathing in, we breathe in oxygen. Oxygen is dissolved in the blood. So that raises the solute in the blood. When our cells do aerobic respiration, one of the byproducts is CO2. Okay, again, when CO2 is dissolved in the blood, that raises the solute level in the blood. Mucus, saliva. So this is secreted by cells. So when cells secrete these into our body fluids, again, that raises the solutes in our body fluids. Solute loss, some examples are urinary excretion. So when we urinate, of course, in addition to water, we lose solutes as well. Respiration, so when we breathe out, we're breathing out CO2. That gets rid of some solutes from our blood. And again, sweating in some species, not only do we lose water, but we lose some electrolytes that are in our sweat as well. Okay, so what we're looking at here, this is the urinary system. So here are the kidneys. So we have two kidneys. Sitting on top of the kidney is our adrenal glands. Okay, our adrenal glands, these are glands that secrete important secrete important hormones. Here's our urinary bladder. So the kidneys are connected to the urinary bladder by two tubes called ureters. And finally, we have the urethra down here. So the urethra is where the urine can be excreted once the urinary bladder is full and needs to be emptied. Okay, so let's take a little bit of a closer look at the kidney. So here's the kidney, and we can divide the kidney into two parts, the cortex and the medulla. Okay, so the cortex is the outer part. So I'm going to draw a line right. Okay, so this is approximate, but everything on this side of that line that I just drew is the cortex of the kidney. Everything in the center here, that's the medulla of the kidney. Okay, so we have the cortex, we have the medulla, we have the renal artery and the renal vein. So the renal artery carries blood to the kidney. The renal vein carries blood away from the kidney. And then we have the ureter. So again, the ureter is the tube that connects the kidneys to the urinary bladder. Renal capsule. The renal capsule, this is dense, irregular connective tissue that surrounds the kidney. All right, let me erase that line that I drew. There we go. Okay, so let's take a little bit of a closer look at the structure of the kidney. Okay. So down here, so again, there's a line drawn that approximately shows the border between the medulla and the cortex. So again, here's the cortex of the kidney, the medulla of the kidney. And notice that running from the cortex to the medulla and back are these little tiny tubes. Okay. These tubes are called nephrons. Nephrons, N-E-P-H-R-O-N. Nephrons are actually the functional units of the kidney. So this is where all the work of filtering the blood, of concentrating nitrogen waste down, and osmoregulation as well. That happens inside these little tubes called the nephrons. Okay. So if we kind of blow up this little section, so here we can see, so here's the cortex of the kidney here. Here's the medulla of the kidney down here. And you can see these little tubes or nephrons run from the cortex down into the medulla. Okay, then they loop around and come back up into the cortex of the kidney. Okay? And again, these little tubes, these nephrons are very important. These are the functional units of the kidney. This is where all of the excretion of nitrogen waste into the urine takes place. Okay, nephrons. Nephrons are the functional units of the kidney, like I just said. They are the structures which accomplish osmoregulation and nitrogen excretion by filtering the blood. There are about, so there's, you might have noticed in the drawing, there's lots of these little tubes. So there are about 1 million nephrons in each human kidney. So 1 million nephrons times 2 kidneys, that means, assuming we have 2 kidneys, that's 2 million nephrons for each individual human. Okay, each nephron is partially located in the cortex and partially located in the medulla. Okay, so looking here, so we can see in lighter brown here is the cortex of the kidney. Darker brown, this is the medulla. Over here, the dividing line is this red line here. So here's a nephron. The nephron goes from the cortex down into the medulla, okay, then back up into the cortex, okay, and then it drains into this collecting tubule here. Okay, so here's another, just another drawing of a nephron. So again, nephrons, so we really can't see the dividing line between the cortex and the medulla. I would approximate it to be right about here. Okay, so again, here's the start of here's where the nephron starts, loops down into the medulla, back up into the cortex, and then empties into this collecting duct here. Okay, let me direct your attention to the whiteboard. And before we start talking about the nephron function, let's talk about the parts of the nephron. Okay, so let's start over here. At this end of the nephron, the first structure that we see is a structure called the glomerulus. Okay, that's Glomerulus. The glomerulus is actually a ball of blood vessels in which the blood's under very high pressure. So these are blood vessels under very high pressure. Now you can see there's a structure that surrounds the glomerulus. The structure that surrounds the glomerulus, this is called the Bowman's capsule. Okay, now this section of the nephron that connects to the Bowman's capsule, that's in the cortex of the kidney. Let me draw a, actually let me use a different color. So again, the division between the cortex and the medulla is kind of arbitrary. I'm going to approximate right about here. Okay, so everything above the red dotted line is the cortex of the kidney. Everything below is the medulla. This section that connects to the Bowman's capsule that's in the cortex of the kidney, this is called the proximal tubule. The proximal tubule connects to this part that goes down into the medulla of the kidney, then back up to the cortex. This is called the loop of Henle. Okay? And this is the structure that's so big in those kangaroo rats that live in the desert. Okay, so this is the loop of Henle. Let me just stop the share to make sure everybody can see this. Okay, yeah, looks like everybody can see that pretty well. Okay, so again, this structure here is a loop of Henle. We can divide the loop of Henle into two parts. We have the descending limb of the loop of Henle, and we have the ascending limb of the loop of Henle. So over here on this side, this is the descending limb of the loop of Henle. Over here on this side, where it's going back up into the cortex, this is the ascending limb of the loop of Henle. Okay, so descending limb, and I'm in the way here, ascending limb over here. Okay, so as we go back up into the cortex, this part here that connects to the loop of the ascending limb of the loop of Henle, and this part here, this is called the distal tubule. Okay, and the distal tubule connects to this part here, which is called the collecting duct. Okay, and again, the collecting duct, just like the loop of Henle, goes from the cortex of the kidney down into the medulla of the kidney. Okay, so those are the parts of the nephron. Definitely know these for the upcoming quizzes and for the test next week. So again, just to go over the parts of the nephron really quickly, we have the glomerulus over here, which consists of blood vessels under high pressure. Surrounding the glomerulus, we have Bowman's capsule. Bowman's capsule connects to the proximal tubule. Proximal tubule connects to the loop of Henle, which goes down into the medulla of the kidney, then back up into the cortex. Okay, we can divide the loop of Henle into two parts. We have the descending limb of the loop of Henle, where we're going down. When we turn and we're going back up into the cortex, we have the ascending limb of the loop of Henle. Ascending limb in the cortex connects to the distal tubule. Distal tubule connects to the collecting duct. Okay, and again, the collecting duct is going to go from the cortex down into the medulla, just like we see at the loop of Henle. Okay. Question from Brooklyn. What is across from the distal tubule? Do you mean right here? Okay, this is the collecting duct. Did that answer your question, or are you asking about something else? Brooklyn, did that answer your question? I meant the proximal tubule, but you answered earlier. Okay. All right, so again, those are the parts of the nephron. And please do know this for the quizzes and the upcoming exam. Let's go back to the PowerPoint. Okay, so now that we know all the parts of the nephron, let's talk a little bit about nephron function. So at the glomerulus, high pressure forces water and most small solutes out of the blood non-selectively. So basically what the kidney's jobs, well, here are your kidneys. Mostly what the kidney's jobs are to filter our blood, okay? And it's gonna filter out any nitrogen waste from our blood. So at the glomerulus, water and small solutes are forced out non-selectively. What's that mean, non-selectively? That means everything, okay? Urea, of course, that's nitrogen waste we want to get rid of. Ammonia, okay, any ammonia that hasn't been converted into urea yet, that gets forced out, along with everything else, glucose, amino acids, nutrients, good stuff. So everything is being forced out non-selectively, whether it's something good that we want to keep in our bodies or something bad that we want to get rid of. Okay? So at the glomerulus, high pressure forces water and most small solutes out of the body non-selectively, okay, both good and bad, everything out. Most filtered water and solutes are taken up into the lumen of the Bowman's capsule. Okay, so that structure of the Bowman's capsule that I showed you on the board, this is going to collect the water and the solutes forced non-selectively out of the capillaries that make up the glomerulus. Okay? So now at the proximal tubule, solutes to be kept in the body, nutrients and some sodium chloride are transported out to be reabsorbed by paratubular capillaries. Okay. So what's happening here, so remember at the glomerulus, all solutes were forced out non-selectively, okay, both the bad stuff and the good stuff. At the proximal tubule, we're going to transport the good stuff back into the body. Okay, so nutrients, sodium chloride, those are going to be transported out. So we're over here. Okay, sodium chloride, nutrients, some water. These things are going to be transported out and reabsorbed by the body. Okay, and reabsorbed by paratubular capillaries. Now this is, you guys might not be familiar, this is probably a new term for you. Paratubular capillaries. Capillaries are very small diameter blood vessels. Okay? In fact, they're the smallest diameter blood vessels in our whole body, okay? Paratubular just refers to around the nephrons. Okay, so when we talk about paratubular capillaries, we're referring to very small blood vessels that surround the nephron. Okay, because these blood vessels are very small in diameter, nutrients, solutes like sodium chloride can diffuse into them very easily. Okay, and then they'll be carried away to the rest of the body. Okay, so paratubular capillaries, these are going to absorb the nutrients and other solutes that are transported out of the proximal tubule and then carried away to the rest of the body. Okay. And I see we're out of time, so unfortunately we'll have to stop here. When we come back Monday Monday's a review session. So actually this is the last material that you'll be responsible for on Wednesday's exam. Again, look for quiz number 18 to be posted later today, sometime around noon. Watch for that study guide to be posted a little bit later today. Please look it over before Monday morning. Monday morning's our review session. So on Monday, I will just answer any questions that you have on anything that we covered since test number 2. Okay? All right, so we're out of time. Unless there's any questions, I'm going to end the Zoom meeting here. And actually, all the students today are Zoomers. Nobody showed up in person in class. So if there's no questions in the Zoom chat, I'm going to end our meeting today. Oh, here's a question from Nathan. Hey, Professor, are you still going to start doing quizzes Monday? I haven't really talked to the class about that. I'll tell you what, let's talk about that on Monday. If you guys want quizzes 3 days a week, I'll be more than happy to do it. Again, that'll give you more drops and more opportunity to max out your quiz scores. But let's put that on the back burner till Monday. We'll talk about that Monday. All right? If there's no other questions, I'm going to stop the meeting. So you guys have a good weekend, and I'll see you Monday morning. Bye.